Hello, everyone, and welcome to OncoDaily. I'm Amalya Sargsyan, medical oncologist and vice president for research and intelligence at OncoDaily. And today I have the greatest honor of welcoming Dr. Jennifer Buell, who is the CEO of MiNK Therapeutics. Dr. Jen, thank you for accepting our invite and being with us today.
It's such a pleasure, Amalya. Thank you very much. And thank you for what you do at OncoDaily to educate the world on novel and innovative therapies. I really appreciate it. It's nice to be here.
Thank you. It's a collaboration, and we're always happy to give the voice to the new science. And let's together discuss what we have new in this field. And before we just go on, can you briefly introduce yourself and your role at MiNK Therapeutics?
I'd be happy to. Jennifer Buell. I have been involved in developing novel therapies for patients with cancer and other inflammatory conditions, including some viral complications, for now over 28 years. I have been a big part of the work that has been done at our parent company, Agenus, as we've advanced a pipeline of substantial antibodies forward. What we were realizing, though, is despite some of the advances we could bring to patients with cancer, there is what we were observing is that in patients who were having an inadequate response or a less than optimal response to some antibody-based therapies, including T-cell engagers and antibody-drug conjugates, part of the challenge has been an inadequate immune system. And at MiNK Therapeutics, I founded the company to try to address this problem. And what we have been advancing is a novel platform using the most highly conserved and immune potent cell in immunity, and that's invariant natural killer T cells, which we'll talk to you about today. I'm the founder, the president and CEO of MiNK Therapeutics, and I'm working with an exceptional team of scientists, manufacturing scientists, discovery scientists, and clinical scientists to bring some of these therapies to areas of great unmet need.
Thank you for sharing this with us. Indeed, in these days, we see more and more novel therapies coming, and especially in those fields when there are addressed unmet need, even with novel CAR T-cell therapies and so on. We're not there yet, so we need something novel. And we're very happy to have this chance of discussing today what seems very novel and very different from what was already there. And in the meantime, very similar to what they do, but we'll discuss nuances together. So can you say a bit more on what is iNKT cell therapy and how this differs from traditional immunotherapy? So what is it?
Absolutely. A really important part that we need to think about is your immune system was designed it's with two arms, the innate immune system, which is that with which you are born with, the adaptive. And that's the immune system that we think most about as we recover from a pandemic. We think about how our immune system has helped us to overcome novel threats that come to us. And we work on the adaptive immune system by making some modifications, both with vaccine therapies and otherwise. iNKT cells, invariant natural killer T cells, are a subset of T cells. They are not NK cells. And these very potent and very rare T cells modulate both arms of immunity, your innate immunity as well as your adaptive immunity. These cells act as first responders. That's how we think about them. And they're conductors of immunity. And I'll tell you what I mean by that. They not only recognize stress signals very broadly across the body and specifically within tissues, but they do not look for just one antigen or just one pathogen. That's really important. What we have learned over the years in treating patients with cancer, as well as patients with autoimmunity and infections, that these single antigen or single pathogen products are limited. In cancer, we see that this disease really overcomes what a single antigen vaccine approach or a single targeted product could do. And essentially, we just see that evolution of cancer. They overcome what we're targeting. What's beautiful about an iNKT cell is it actually is very broadly acting, and it gives us multiple pathways in cancer, inflammation, as well as in infections. And this is with a single unmodified dose. So effectively, we isolate invariant natural killer T cells, we scale them so that we could create billions of cells per dose. Then we administer those cells to patients. And it's a fundamentally different modality than what you have access to today. It's really precision without complexity. And it positions these cells actually as one of the few scalable options for cell therapies that are really capable of moving beyond these niche cancers into really a much higher unmet need and beyond cancer as well, which we haven't seen with CAR Ts or with other immune cell therapies at this time.
Well, thank you for explaining so nicely. I think everyone, patient and scientists, will now understand how this works. And indeed, in cancer, we're learning to work with the microenvironment because tumor is into the microenvironment. Our immunity is very different and very complex. So we have to act on different level and try to get the best way to overcome it. And single antigen cannot be sometimes the resolution with it. And you were explaining it how it works. So it's a single dose administration, and we're creating billions of cells to give the chance to fight the cancer. But and in your reports, it's mostly that it's off the shelf. You usually call it, right? Am I right?
That's right.
So what does it actually mean? So it doesn't require any fabrics, or can you a bit explain about it?
Absolutely. So this off-the-shelf or allogeneic iNKT cell therapy, it really means immediate availability, no chemotherapy. This is a major clinical and economic unlock. Hospitals can infuse the cells within hours, not weeks. They don't have to wait for a patient to come in, isolate cells, manufacture them, and then deliver them weeks later to the patient. These are immediately available when the patient arrives. And we don't need to build any specialized infrastructure. Hospitals do not need any specialized infrastructure. And we're able to leverage our GMP facility in Lexington, Massachusetts to actually just isolate, scale, and produce and ship these cells. What's really important for cancer, specifically, this expands access beyond what's available today. For patients with respiratory failure or complications of lung disease, autoimmune lung disease, idiopathic pulmonary fibrosis is an example, or infections, drug-resistant infections, you need to move quickly, immediately, in fact, actually within hours in many cases, particularly these resistant infections. With our therapy, it's off the shelf, it's available. We send it to the hospitals in advance, so it's immediately available when the patient arrives. It allows treatment at the moment patients are declining and the moment the patient's in need, where current options fail. So operationally, we finally have made a cell therapy that's actually deliverable at scale and can be on site when the patient needs it and at the time that the physicians need to administer it. And we see that in our clinical trials now. We just ship the product to the sites. It's stable, frozen for many years actually now, and we're continuing to collect more data on that. And effectively, all the hospital needs to do is take it out of the freezer, bring it to the patient's room, and they just hang it and use a gravity infusion similar to how you would administer nutrition in any hospital, or plasma for that matter as well.
Wow, that's impressive, because with the novel drugs, sometimes it's a matter of infrastructure too. And it's not only the efficacy, it's also deliverability of the drug. A lot of CAR Ts and cell therapies are very promising, but not all hospitals can afford themselves and cannot have this infrastructure. And this is not applicable for the rest of the world. And this could be a revolution. And this is indeed a very important point. So you're just using it, it's already there, and we have to give the drug if there is an indication. That's, I think, one of the most important points and advantages of these therapies. And thank you for explaining how it works. Hopefully, a lot of part of the world will have access of these kind of therapies and will give the most of the benefit of cell therapies, including these invariant iNKT cell therapies. So, moving on, I know that the data and the trials where the iNKT is tested was in pretreated solid tumors, very resistant population, difficult, hard to treat, exhausted, a lot of lines. Why do you have chosen this kind of tumors, and is it effective? How it's overcoming the challenge? What is your rationale of choosing these difficult to treat populations?
Yeah, thank you. That's a great question. There are two parts to why we selected this patient population. First, it's a great unmet need. And we needed to start with a phase 1 clinical trial that allowed us to not only evaluate safety, but also to identify signals of activity in a population of patients who had nothing else. That defined our initial clinical trial. It also allowed us to get information about what the cells can do in a setting that other more commonly used or standard available therapies were not functional or did not work for patients. We saw really some remarkable findings that allowed us not only to think about how to expand in some specific tumors that have an unmet need, but that are failing what is most commonly used. In some cases, a simple PD-1, anti-PD-1 therapy, which many know is Keytruda or Opdivo or others, and they're very commonly used across a host of solid tumors. But what we observe is that many, in fact, the majority of patients actually progress on those therapies once they're on for a certain amount of time, and then they have nothing else. And in many ways, they progress because they have an inadequate immune response. And what we can do is augment or restore that immune response. And I'll tell you, these solid tumors are immunologically suppressed environments. They're designed in order to survive and thrive. Cancer is such a sophisticated organism, we could say in a crude term. And most T cells, engineered T cells, lose function or they cannot penetrate the tumor. Where iNKTs are intrinsically different is they traffic to the inflamed tissue and they maintain activity in these very hostile environments. And importantly, we do not see that they exhaust. In all of our preclinical tests, they do not exhaust. And what we're seeing in humans is that they don't exhaust. They proliferate when they see the target. They also, once they get to where they need to go within the tumor tissue, they actually recruit a broader immune system. So think about these as soldiers that get to where they need to be, and then they start to release a chemical signal that recruits a big, broad group of T cells and natural killer cells, where you can get a very rapid killing of the tumor tissue, and then a long memory, durable memory response, which I believe gave us the long-term findings that we saw. We were really quite excited and surprised, frankly, to see that so many patients with these very refractory tumors were living a long time, sometimes 2 and 3 years. And in fact, iNKTs delivered as a single therapy, a single dose, in a patient with thymoma, relapsed refractory thymoma, is living now more than 3 years. We similarly saw patients who had failed on an anti-PD-1 therapy after they had exhausted all standard therapy. And in some cases, we saw a complete clinical response by administering the cells in combination with continuing their PD-1 therapy, commercially available PD-1 therapy. This allowed us to see if when patients fail, can you reconstitute their immune system and see a benefit? And we did. We saw a complete clinical response in a patient with testicular cancer who had failed everything else. And subsequent to that, we saw patients with renal cell adenoid cystic carcinoma who had very long durable survival, and some continue to be alive today. Our translational data, we've presented this at some major conferences, show why that may be happening. And what we see is that this product, AGENT-797, really retains something that's called polyfunctionality. And it engages macrophages and dendritic cells. These are very important cells in your immune system. And that allows it to change the immune milieu or the immune system around the tumor. And it makes it really hostile for tumors to continue to grow. And that's when you start to see some real benefit to patients. So we believe that we have defined a path for repeatable efficacy without having to engineer the cells and without having to lymphodeplete. We do not have to administer toxic chemotherapy to lymphodeplete patients, which you do not see with other available therapies. You often have to make room for the cell therapy with other T cell therapies and engineered cells. You love to lymphodeplete. You don't have to do that with iNKT cells.
Wow, that's very impressive. And it seems that you are giving the second chance of immune system to work against cancer. And this can have durability of the response, which is also very important. Thank you for sharing some nice cases. Hopefully these cases will become more and more, especially these stable diseases called tumors, very important. And we are learning it every day that reprogramming and giving a way to work with therapies is really a way to overcome the treatments. You have already mentioned some major results presented at conferences, and I know that your data was also presented at the recent Society of Immunotherapy Cancer Congress. And would you like to highlight anything more from that Congress, though I saw that you mentioned some? If there is anything else.
I mentioned the exciting responses. So initially what we had presented earlier in the year was at the inaugural American Association for Cancer Research. And we revealed some very intriguing clinical and translational findings where we saw that patients who had failed primary therapy for second line for gastric cancer, they then were treated and had failed FOLFOX and PD-1. And some of those patients continued on their PD-1, and we added the cells to that PD-1. And we saw some remarkable responses, clinical responses in those patients. The tumors were shrinking and patients were surviving longer than we expected. What we presented earlier in the year is why that is. And we looked at the tumors before we treated them from a biopsy and pathology. And we saw that there were no immune cells within that tumor, nothing to fight the tumor. Once we administered a peripheral, just a simple IV dose of iNKT cells, we saw an immediate change within the tumor. We saw very effective CD8 T cells and CD4 T cells rushing into the tumor. And we turned an immune desert, as we like to call it, a cold tumor, into something that was very inflamed or very hot tumor. And that's where you start to see the disease elimination. And that association, what we saw is the immune system went in, and then we started seeing the tumor shrink. That led us into a phase 2 that's being funded by outside sources that are quite excited. Stand Up to Cancer is one of the funders for this trial, with Memorial Sloan Kettering's leader, Dr. Yelena Janjigian and Dr. Sam Sataran. They're advancing a phase 2 trial in second-line gastric cancer. We presented some of those the results from our primary trial then later in the year, just now at SITC. We also presented data from a patient who had testicular cancer, as I mentioned, renal cell carcinoma, adenoid cystic carcinoma, and thymoma. And those patients had very long survival. These are patients that you'd expect to see 5 to 6 months of overall survival. And we had a median of 23 months, and some of those patients still alive and being followed. An important thing I did not mention is the safety profile. With iNKT cells, they have a very unique and invariant TCR. It's common in us. And therefore, I can take cells from a healthy donor and I can give them to a patient. We do not see rejection. We do not see graft versus host disease. And in fact, we see that the cells may actually reverse graft versus host disease. And when you looked at the safety data we presented at the SITC conference, you saw a very favorable tolerability profile. No GVHD, no cytokine storm, no neurotoxicity, and that's really rare for a cell therapy. And that has led us now to take this program into next phases in development, into a phase 2 trial that's continuing in gastric cancer, into a phase 1 trial in patients who are undergoing hematopoietic stem cell transplant. We're going to look to see if we could add these cells and ensure that they not only get a better engraftment of the cells, but also they do not have GVHD. They do not develop GVHD. That's what we're hoping. We want to prevent GVHD in this population where nearly half of them progress. And we think we could do so with a very tolerable therapy, single administration. And when we administer these cells, we are actually reconstituting or restoring about 20% of a patient's immune system. And we see that in the immune system, we're really the first to show that, like other organ systems, like a liver, if 20% of your liver is working, you will have a proper function of your liver, similar with kidneys and other organs. And what we're doing is restoring 20% of an immune system. And we're seeing the results. We're seeing that if we do that, we see some really remarkable and unexpected outcomes for patients with a safety profile that allows this product to be tested across a lot of very vulnerable patients at a time of great need for them.
Oh, thank you for this nice summary. Indeed, safety profile is the second concern about after efficacy, because you have to be able to give the drug, to be able to give it safely, and seeing that it has much less side effects than the usual cell therapies, which scares a lot of people and also researchers, and you need to be very prepared with very distinct units and all the side effects in our hand. It's important to see that the safety profile is thinking of good. And you have mentioned that you are planning on the phase 2. Congratulations on that. Hopefully, while our next presentation, we'll have some preliminary results, and already will be a study ongoing. And how do you see the development of iNKT cell next, like in general? Do you see it moving to the more front line to be used in the combination with other treatments? Or you see it as a monotherapy to overcome the resistance of the standard immunotherapies? Just the general overview of your idea of understanding the future for iNKT.
Thank you, Amalya. We're going into the earlier disease setting as opposed to the late line. We've already established the cells can be dosed, that they're tolerable. Patients can receive them without toxicity. And that allows us now to start developing in earlier disease settings, where if you can actually reconstitute the immune system in that setting, I believe we could have major, substantial benefits for patients. So we will be going into the earlier disease setting, and you're going to see some of the upcoming external funded trials that are moving into that direction, and patients with colorectal cancer, and also patients with germ cell tumors. So this gives us a big opportunity to do this in cancer. Similarly, we're going to build on the phase 1 data in autoimmune conditions with GVHD, and that's also funded by 2 big programs. One is an NIH-funded and DoD-funded preclinical program, and then a privately funded philanthropic program for the clinical study that will be announced as a first-in-man is starting very imminently. That allows us to, and that's in GVHD. So a phase 2 trial in cancer, a phase 2 trial externally funded in patients to prevent graft versus host disease, and finally a MiNK-funded study in which we're going to build on our findings of respiratory distress. And we're going to help patients. And it really, as we start to think about the winter is coming, and we see that these infections and other complications from infections complicate the lungs, and the lungs look different post-COVID. We're seeing younger and younger patients succumb to multi-resistant organisms. We've shown that, and we will be publishing data on this, that we could really help to restore the immune system and eliminate some of these really complex viruses and bacterium, Pseudomonas aeruginosa, other fungal infections. And we believe that given the scale and the very affordable cost that we've been able to manufacture these cells at, we could give physicians on the front line of these critical illnesses an opportunity to actually deliver a broad-acting, anti-pathogen and effective therapy to patients who need them most, that will allow them to clear these pathogens from their lung tissue, as we saw in the phase 1 study, and get them out of the ICU very quickly. We've already demonstrated this. We've published it in Nature's Communication in 2024, and we're going to build on that data in a study to start very quickly, a phase 2 study in patients with severe respiratory distress. And that more details on that program will be announced very soon. Very exciting time for us right now.
Yes, indeed. You're advancing in different front lines.
Yes. Yes.
Nice to hear that. Actually, sometimes it's difficult to understand, but this is when you are working with immune system. So it's not only cancer. It could be very different diseases, all very different, but in a way you are helping your own immune system to fight against. So this is the advantage of the method. Possibly it's not just giving the drug, but also learning how our mechanisms work and helping them. Indeed, very promising, and I'm sure by the next time, hopefully we'll have even more fronts to fight for. Congrats on everything you have achieved. This is tremendous work. I can imagine with a huge team, a lot of researchers, scientists, patients involved in it. And thank you for explaining how it works and all of this data, summarizing and putting up together. It was very interesting. I'm sure all our listeners would love to hear this. whether it's for themselves, for their loved ones, or as a researcher to do another trial. Would you like to give some message to your patients or the researchers who are listening and want to know about iNKT cell therapies?
Sure. I have to express my sincere gratitude to our patients, very courageous patients who and their families who are willing to take a chance and participate in research. And for those patients, we are immensely grateful because we couldn't do what we're doing without them. And we work so hard to try to benefit them. And by participating in research, it really takes an enormous gift. It's a treasure that helps us to move these cells to a broader patient population, to more people who can benefit. And so for that, we are incredibly grateful. And to our physicians who also have not only the courage, the intuition, the skill to help us move these cells forward, we're just immensely proud and grateful. And I think for those scientists and developers who are thinking about how do we expand the benefit for patients, these iNKT cells, AGENT-797, is really designed to just restore immune control when it really matters, really most. It's in hard-to-treat cancers and in life-threatening pulmonary diseases. These cells can be delivered quickly. They are accessible. They have a safety profile that allows treatment earlier in the disease course. And for clinicians, it provides really a new immunologic lever where existing therapies fail and or fall short. And for patients, it represents a scalable and next-generation immune medicine, really designed to meet these real-world needs for them when they need them most and when they're most vulnerable. So I'm so thrilled to have the opportunity to talk with you and your team and continue to look for the work that you're doing. And I appreciate the time that you've spent in helping us to educate on these really novel and exciting therapies at a time that's most exciting for MiNK.
Thank you for accepting our invite and being our guest. It's our pleasure. We want science to be accessible to everyone and to give voice to everyone, especially when we are seeing such good results. And this was our pleasure to host you. And stay tuned for more updates. Thank you so much.
Thank you.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.